Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry mentor. In this video, we are going to discuss nitrogen balance and the metabolism of amino acids as per their nitrogen skeleton. So, let's start up with the discussion. Metabolism of amino acids as per their nitrogen skeleton. Now see, basically how do we define an amino acid? We all know that amino acids are the molecules, yeah, you can say are the macromolecules which are forming a protein, and they have one amino group and one carboxylic group. And this becomes the generalized structure of all the 20 standard amino acids, right? Now just looking at this structure, I can say that they differ in R group. So I can say that R group will be different for all the 20 standard amino acids. So if we talk about the metabolism, we all know that metabolism comprises of two parts. Catabolism is there, anabolism is there. Catabolism means breakdown of something, anabolism means synthesis of something. So first of all, these amino acids, because they are forming proteins. So what happens is when the proteins undergo breakdown, they are going to form these amino acids, and these amino acids metabolism will be decided as per their nitrogen skeleton and as per their carbon skeleton. Because this particular structure will be the same for all the 20 standard amino acids. This will be same. And amino acids are the only macromolecules, I would say, because they are forming the proteins which have this amino group in them. And this amino group is given out from the body, it is expelled from the body in the form of urea. Okay. So, if we talk about these amino acids, their metabolism, first of all, if we talk about the nitrogen skeleton, right? So, this nitrogen which is present in amino group, first of all, it will undergo transamination. First of all, the group will be transferred, then the group will be removed, which is deamination, and in the end, this amino group is going to form urea, or it will undergo urea cycle and it will be expelled from the body. And that is why we say we human beings are what? We are ureotelic in nature. Okay, so this is very important to understand. Now coming over to this, there exists a nitrogen balance which is very important to understand. Now what is this nitrogen balance? Because I told you amino acids are the only one, because if you see the structure of carbohydrates, if you see the structure of lipids, nitrogen is not present as the main one. Okay, derivatives are there, but nitrogen is not the major component. But if you look at the structure of this amino acid, all the 20 standard amino acids will have the basic structure. So this nitrogen balance is there, and always remember nitrogen balance can be 0. It will be 0 in a healthy individual. Or we can say 0 means when protein synthesis, the rate at which the proteins are synthesized is equivalent to the protein degradation. So nitrogen balance is 0. Then this nitrogen balance can be positive, rather it will be positive. So nitrogen balance can be positive when the synthesis is more than breakdown, right? And this nitrogen balance is positive mainly in the growing individuals, you can say the youth people. So I can say in the young people and the pregnant women where the requirement of protein is more, pregnant and lactating mothers. Right. Then it will be negative. Now, if we talk about when it will be negative, basically when synthesis is less than the breakdown, or protein degradation is more than synthesis. And this only occurs in any injury, or most commonly it occurs in diabetes mellitus. So in diabetes mellitus, and we can say in any injury, renal injury, or you can say, then there will be breakdown of the proteins. So this is very, very important to understand, and nitrogen balance is basically only present in the amino acids which are forming the proteins. Now, if we talk about what is the source of the amino acids, so I can say that these amino acids, from where they are coming? They are essential, means they have to be provided from the diet. They are coming from the diet, or they are coming from the proteins breakdown. Right? And this amino acid, first of all, you can say that they will be required for the synthesis. So the pool of amino acid will be required for the synthesis of porphyrins. Like heme is an iron porphyrin. Amino acids are required for the synthesis of purines and pyrimidines. Right? Then we can say amino acids can be converted into glucose, and amino acids can be expelled from the body by forming urea. So whatever factors which are giving rise to this amino acids or nitrogen balance, that should be equivalent to the factors which are utilizing these amino acids. Okay, so that the metabolism term is justified. Means you can say catabolism plus anabolism is equivalent to the metabolism. Whatever energy is required for the synthesis of something, that energy will be equivalent to the degradation of something. And one very important thing about proteins is that these proteins stay in our body till the last. Means prolonged starvation. Say if we are starving for 4-5 days, proteins are the ones which will be giving energy to the body. Okay. So now we are going to study the metabolism as per nitrogen skeleton first of all. So the first process here is transamination. Right? Now, transamination, as the word says, there is transfer. Kiska transfer? Obviously the amino acid, because here we are basically only focusing on the amino group. So, transfer of an amino group to whom? To a keto acid. So, what will be produced? To form a new amino acid and a new keto acid. Right. So, this becomes the definition of transamination. Trans means transfer, amination means amino group. So, the thing is that what is the enzyme required for this? So, the enzyme required for transamination is obviously transaminases or aminotransferases. And the most important point here we have to remember is the coenzyme required for transamination is vitamin B6 or PLP. So, the enzyme is transamination and the coenzyme is vitamin B6 or PLP. Now, what are the examples of transamination? So if we talk about the examples, there are two main enzymes which are diagnostically also very, very important. Now they are, one is ALT, which stands for alanine transaminase. Alanine transaminase. So this is, the amino acid here is alanine. So what would be the reaction? This alanine will combine with a keto acid. That is what the definition says. Keto acid here is alpha-ketoglutarate. It is going to form a new amino acid which is glutamate, and a new keto acid which is pyruvate. Okay, and always remember it is reversible. So I can say that it is a reversible reaction. Right. So alanine ka corresponding keto acid is pyruvate. Okay. And clinically it is also known as SGPT. SGPT stands for serum glutamate pyruvate transaminase. And if we talk about the significance of this ALT or SGPT, it is a intracytoplasmic enzyme first of all. It is a cytoplasmic enzyme. And if we talk about the, you can say, significance of this, it is a marker of liver diseases. Very important. Marker of liver diseases. It always increases in non-alcoholic fatty liver diseases, right, and it will increase when there is acetaminophen toxicity also. Right. So this is alanine transaminase. Second enzyme is AST. This AST stands for aspartate transaminase. And if we see the reaction catalyzed by this, aspartate is the amino acid. The keto acid will remain the same. It is alpha-ketoglutarate. Since transamination is reversible, the new keto acid, the new amino acid first of all will be glutamate, and the new keto acid will be oxaloacetate. And glutamate and oxaloacetate are there. That is why clinically it is known as SGOT, which stands for serum. S stands for serum glutamate oxaloacetate transaminase. Right? And if we talk about the occurrence of this, so AST or SGOT, it is partly cytoplasmic, partly mitochondrial enzyme. So it is cytoplasmic as well as mitochondrial enzyme. Mitochondrial enzyme, right? And if we talk about the significance of this, it is a marker of liver diseases also, and it is a MI marker also, or cardiac marker. Then it increases, always increases, you can say, in liver cirrhosis. It will increase. Alcoholic fatty liver diseases. And it also increases in liver neoplasia. Right, because there is actually a ratio. We call that ratio as AAR or AST to ALT ratio. So it is more than 1 or it is less than 1. This is how we categorize it. Okay. So these are the examples of transamination. Now, I told you there are 20 standard amino acids. So always remember, out of 20 standard amino acids, only 17 undergo transamination. Right? And the 3 amino acids which do not undergo transamination, they are, again there is a mnemonic for that, 3 amino acids not undergoing transamination are, the mnemonic is polythene, where P stands for proline, L stands for lysine and T stands for threonine. So these are the 3 amino acids which cannot undergo transamination. There is, in some books it is mentioned serine also. So apart from this, all the amino acids are going to remove, are going to first of all transfer their amino group, or they will undergo transamination, right? So if you have to highlight it, the most important thing is the coenzyme required for transamination. Always remember, it is B6 or PLP, pyridoxal phosphate. Now once this group is transferred, it has to be removed, because our ultimate goal is what? We have to eliminate this nitrogen which is present in the form of amino group. So next process is deamination. Now as transamination means transfer, deamination will mean removal of the amino group. Right? Now this deamination can take place in 2 circumstances. One is in the presence of oxygen. So if it takes place in the presence of oxygen, we call it as oxidative deamination. And if it takes place in the absence of oxygen, it is known as non-oxidative deamination. Right? So we can say that in the presence of oxygen and in the absence of oxygen, whatever is there, amino group is removed. So if we talk about oxidative deamination, it is clear from the word itself, oxygen is required, and this is in the presence of auto-oxidizable flavoproteins. Well, flavoproteins. And the enzyme here is L-amino acid oxidase, because this we have already discussed in the chemistry that amino acids are also present in D form and L form, depending upon the position of the NH2 group. So what happens is, this L-amino acid, right, it will first of all undergo oxidation, forming a intermediate. Now why I have written square brackets? Because intermediate is generally unstable, and it will dissociate, it will finally release this amino group and it will be converted into the keto acid. Now since it is oxidative deamination, so the enzyme here will be L-amino acid oxidase. And I have mentioned this auto-oxidizable. Auto-oxidizable means FMN is reused as a reductant here, and this is the only reaction in our body which requires FMN. Now what is FMN? It is flavin mononucleotide, which is the active form of vitamin B2. So FMN will be converted into FMNH2 like this, and this is connected with the degradation of hydrogen peroxide. That is why it is auto-oxidizable like this. Okay. So this is oxidative deamination, and non-oxidative means we can say without the presence of, in the absence of oxygen. So I can say histidine can be converted into urocanate. There are so many which we will be discussing in the metabolism of the amino acids itself. So histidine can be converted into urocanate. Okay, but here oxygen is not required. Now coming over to this, so I can simply say one thing, that all the amino acids in general have their respective keto acids. So amino acids can be converted to keto acids and keto acids can be converted back to amino acids. So if I talk about alanine, the corresponding keto acid of alanine is pyruvate. Aspartate, the corresponding amino acid is oxaloacetate. Glutamate, the corresponding amino acid is alpha-ketoglutarate. So you can just summarize everything that TCA cycle is actually connected with the amino acid metabolism also, as you can see here. Okay, so this is deamination. Now the most important point which we have to see here is, if you talk about any transamination reaction, we can say that transamination is actually occurring between these two. Rest, if you see, everything is different. I mean, like the amino acid is different and the keto acid is different, but the new amino acid is the same and the first keto acid is the same. So what inference we get from this? The inference we get is all the transamination reactions are going to increase the pool of glutamic acid. Now the question is, glutamic acid itself is an amino acid. So what is the point of the metabolism? That means glutamic acid has to undergo altogether a different process. And that is what exactly happens. So what happens here is that this glutamate or glutamic acid, or I would say, what is the fate of glutamate? Fate of glutamate is that it has to undergo two processes at the same time, because the transamination, if I have to explain in simplest language, transamination is leading to increased pool of glutamate. So this glutamate will first of all transfer and immediately remove. So you can say that it will undergo a coupled process, which is transdeamination. Mane transfer followed by removal of the amino group, and this glutamate will be ultimately converted into alpha-ketoglutarate. So this becomes the fate of glutamate. Now the question is, what is the enzyme required for that, where this reaction occurs and how this reaction is regulated. So if we talk about this reaction takes place in the liver and the kidneys. Liver and the kidneys, right. Second important point here is that the enzyme, very important, the enzyme here is glutamate dehydrogenase. Glutamate dehydrogenase. Now since dehydrogenase is required, or you can say that means oxidation is there. Okay, because whenever we write dehydrogenation, it means oxidation. So here I can call this process as oxidative deamination also, because ultimately we have to remove the amino group, na? So since this reaction is taking place in the presence of dehydrogenases group of enzymes, so it will be oxidative deamination, and commonly we can call it as transdeamination. So this glutamate dehydrogenase is a zinc containing enzyme. It's a tetrameric enzyme. And the reaction is again simple, that this glutamate will, in the presence of again glutamate dehydrogenase NAD to NADH plus H positive, or NADP to NADPH. It will be converted into an intermediate first, and this intermediate will be then converted into alpha-ketoglutarate, giving out amino. And the name of the enzyme here will be glutamate dehydrogenase. Intermediate mein again I am just not mentioning the name of the intermediate, I am just writing in square brackets ki that what is the fate actually. So fate is glutamate will undergo simple dehydrogenation, it will remove this amino group and it will be converted into alpha-ketoglutarate. Okay, and that is why it is known as oxidative deamination or it is known as trans-deamination. So I can simply say that now the question is amino group is released. Okay, so as per nitrogen skeleton, what we have done is in the body transamination means transfer of amino group is there, then removal of amino group is also there. But the tragedy is this amino group is present in the form of ammonia. And this ammonia cannot move in the free form. This is again very important. This ammonia will never move in the free form. Why? Because it is going to disturb the acid-base balance. Why? Because you can see NH3 positive. It's an alkali. So it is going to result into alkalosis. So this ammonia needs to be removed from the body as early as possible. And how it is removed? This is removed by the formation of urea. So urea cycle takes place. Now the question is, ek to why this ammonia is toxic, one thing, and second is how this ammonia has to be transported from the brain to the liver or from the muscles to the liver, because urea cycle can occur only in the liver, neither in brain nor in the kidneys, because obviously due to the absence of the important enzymes required by urea cycle. Okay, so first of all we are going to study that this transamination and deamination is leading to the synthesis of release of these ammonium ions, and this ammonia cannot move in free form. Okay, so increased concentration of ammonia will lead to alkalosis or metabolic alkalosis, right? And this will have the worst effect on the brain. And what it will lead to? It will lead to 𝗵𝗵𝗵𝗵𝗵 will be there. Then slurring of speech will be there. It will be difficult for the person to speak much. And blurring of vision will be there. Right? So these are technia will be there. So many things will be there whenever there is increased concentration of this ammonia, because it is leading to metabolic alkalosis. That means what is the biochemical reason behind that, or what is the rationale behind that increased concentration of ammonia? So what happens here is that, see, deamination is the process of removal of amino group. So if amino group is added, it will become amination. D mane removal, addition mane amination. So whenever there is increased concentration of this ammonia, see, there will be a series of reactions occurring in the body. सबसे पहले alpha-ketoglutarate का concentration will decrease. Why? Because alpha-ketoglutarate will be converted into glutamate. So basically the concentration of glutamate will increase. But when the concentration of glutamate will increase, it will be converted to glutamine. So again the concentration of glutamine will increase. So you can see just by one thing, increased concentration of ammonia is leading to decreased concentration of alpha-ketoglutarate, increased concentration of glutamate, increased concentration of glutamine. Now what will happen if the concentration of alpha-ketoglutarate decreases? That means TCA cycle will decrease because it is the major intermediate. Then ATP will be depleted. So ATP depletion will be there. So obviously the brain is going to suffer because TCA cycle plays a very important role in the brain. Coming over to glutamate, this glutamate will increase concentration of glutamate. It will be diverted to form GABA. Gamma amino isobutyrate, which is an inhibitory neurotransmitter. So it is going to affect CNS. And increased concentration of glutamine will retain water and it will lead to cerebral edema. There will be retention of fluid in the brain. So I can say that means this toxic ammonia will lead to depletion of ATP, lead to cerebral edema, lead to increased concentration of glutamine, increased concentration of glutamate. That is why ammonia is highly toxic. Okay, so what is the moral of the story? If something is toxic, it has to be reported. It has to be excreted out from the body. And ammonia can be excreted only by forming urea, which is occurring in the liver. So now the question is how this ammonia is transferred from the brain to the liver and from the muscles to the liver. So what is, I would say, fate of this toxic ammonia? So what happens here is that from the brain to the liver, how it will be transported, first thing, and from the muscles, how it will be transported. So first thing is, from the brain, glutamate will take this toxic ammonia and it will be converted into glutamine. And this is a very important reaction. The name of the enzyme here is glutamine synthetase. So whenever synthetase is there, automatically ATP is required. We know that synthetase, this we have already discussed, and this reaction is irreversible. So by an irreversible reaction, glutamate is converted into glutamine. So I can say glutamine is actually the transportable form. So this glutamine will take this amino group or ammonia and it will hand it over to the liver, where it can undergo the formation of urea. And once it handovers this, it is converted back to glutamate. And here the enzyme is glutamine hydrolase, or you can say glutaminase. So this reaction, first of all, is irreversible. Second important point is ATP is required. So it is going to form, simply you can say glutamine, or glutamine is the transportable form of this toxic ammonia from the brain to the liver. Then second comes from the skeletal muscle to the liver. Now how this transport takes place? This transport takes place by alanine. So what happens here is, say if this is the liver and say this is the skeletal muscle. So what is happening there? We can simply say that pyruvate is being converted into alanine by simple, already discussed this reaction. Alanine and pyruvate are the, basically you can say, keto acids and the amino acids. So this alanine will enter inside the liver, where it will be converted back to pyruvate, releasing this ammonia. And pyruvate, there are so many different uses of pyruvate. It can be used for glucose synthesis, it can be used for oxaloacetic acid synthesis, so many are there. Okay, so from the skeletal muscle to the liver, alanine to pyruvate is there. Right? So ultimately what we get is, we get this ammonia in the liver, where now it will undergo urea cycle. So in a simplest language, I can say that as per the nitrogen skeleton, transamination, deamination, finally it leads to the formation of urea, right? So this is metabolism of the amino acids as per nitrogen skeleton. I hope this video is clear to all of you. In the next video, we are going to study how this urea is produced in the liver, what are the steps and what are the inborn errors of metabolism associated to urea cycle. So that's all in this video. Thank you so much.